no longer there. May his soul rest in peace. And as a mark of respect, let us observe 1 minute silence. Okay, thank you. Now, this is the first of a series of NMRS webinars being organized from the office at Bangalore. The idea came up from Jitendra, Professor Atreya himself, Vivek, and Surya Prakash when we realized that the COVID pandemic is not going to let us hold it for all of us to meet in Pune. And therefore, it was a suggestion from Jitendra and Atreya said, why not we conduct online meetings? In fact, when this idea was proposed, many people said it's a wonderful idea. There is a great welcome sign that we should do it, we ought to do it. And if it is a regular feature, we could even include tutorials for youngsters. The plan of this webinars is to have 2 30-minute meetings on Tuesday. The timings, of course, will depend upon the speaker's convenience and can vary anywhere between 6 p.m. and 8 p.m. Occasional tutorials in important topics also will be covered, depending upon demand. We dedicate also, I want all of you to listen, that we dedicate this NMRS webinars to the memory of Professor Hanudata Sastri Atreya. Now I'm going to thank Professor Rammurthy for agreeing to be our first speaker in the webinar series. A young man who hails from India and has done extremely well, and he is doing guns in solid state NMR. Let me take this opportunity to just briefly mention something about Professor Rammurthy. He is currently Robert Parry Collegiate Professor of Chemistry and Biophysics at the University of Michigan, Ann Arbor, USA. He obtained his PhD from IIT Kanpur after completing his graduation and post-graduation from Madurai Kamaraj University. He has obtained several accolades, including 2015 Hans Fischer Senior Fellow, Technical University of Munich, 2012 Rockham Faculty Recognition Award, 2009 American Association for the Advancement of Sciences Fellow, 2009 Japan Society for Promotion of Science Fellow, 2009 again Wilsmore Fellow, University of Melbourne, Australia, 2009 Distinguished Visiting Professor, Max Planck Institute at Mainz, 2008 NSF Career Development Award, 2008 Distinguished Visiting Professor, Kyoto, and Distinguished Visiting Professor at Osaka University. He include the structure and dynamics and function of macromolecules with a special interest in membrane-associated peptides and proteins. The specific biological systems investigated by him are cytochrome P450, P450 reductase, cytochrome B5, misfolded amyloid proteins, amyloid beta and IAPP, and antimicrobial peptides. Other systems that are being investigated in collaboration using ENMR techniques are bone and nanomaterials. He has 2 books to his credit: MR Spectroscopy of Biological Solids, 2000 published by the Chemical Rubber Company, and Thermotropic Liquid Crystals Recent Advances, edited 2007 by Springer, publisher by Springer. So with this short introduction, I thank Rammurthy for accepting our invitation to give a talk. And Rammurthy, take it away.
Thank you very much, Professor Subramanian, for that very kind introduction. I'm really humbled. I'm pleased to be part of the NMR Society of India. Let me see if I can share my screen. Can you see my slides?
Yes, sir. Yeah.
Okay, great. Thanks to the organizers as well. I would like to dedicate this seminar to Professor Atreya. Atreya is a great scientist. I echo completely what Professor Subramanian said about Professor Atreya. Although I met him very briefly, that too in China, I was quite impressed with my very brief conversation with Atreya. So that was brief enough or long enough conversation for me to continue my interaction virtually with Professor Athria. And he also exchanged with me a lot of ideas, a lot of projects are in progress between us, collaborations as well as Indo-US exchange program, and then conference plans and so on and so forth. So it's really saddening to hear that he's no more with us. I give my condolences to his family and his friends and colleagues and his research group as well. So although my interaction with Professor Athria is very brief, but I have been following his work quite some time, mainly because of the postdoc here in my lab, Tirupati Ravula, right here. He's the one here with Professor Athria. He joined my lab about more than 5 years ago. He's simply outstanding. He's one of the exceptional scientists that I have seen to come out of India, very creative. So that gives an example of how great as a mentor Professor Athria has been, and a great teacher, and so dedicated in training the younger generation. So in addition to Thiru, I also recruited another postdoc, Krishna Arjuna Bankla, right here, along with Professor Athria. He's also working really well. So these two guys are really a backbone of my lab, along with others, and I'm able to survive in this cutting edge research. So what I thought I would like to do today is to focus on the dynamic interaction between proteins in the cell membrane environment, with a specific focus on what NMR can offer here in probing at high resolution. There are two projects dedicated to addressing biological questions in two different areas. One is on understanding high resolution details of structural interaction between P450 enzyme and with its redox partners in membrane. It's a detox enzyme. It metabolizes more than 70% of the drugs in the current market. Second area, which is a very large area, where protein-protein interaction drives the protein aggregation or protein misfolding and so on and so forth. My focus is specifically in the membrane arena. So these protein aggregation is implicated in numerous devastating diseases, including Alzheimer's, phase separation, and more. So here are the topics for today. I will introduce briefly NMR approaches specifically to probe high resolution monitoring of amyloid aggregation in real time. NMR is a very slow experimental technique, but powerful. If we carefully prepare the sample and select the NMR techniques judiciously, we should be able to obtain powerful insights into the misfolding pathways and then nucleus formation and so on and so forth. I will also show an example of how we can probe micelles-like aggregation of our oligomers of iron-to amyloid polypeptide, whose aggregation is implicated in type 2 diabetes. Then I'll briefly introduce a solid-state NMR approach, magic angle sample spinning approach, which is well known, how we can use it to characterize amyloid beta oligomer. And then I'll end with some of the novel nanodisk, which I talked about in the last week, to characterize amyloid oligomers. So what's an amyloid? Amyloid is most of the proteins are intrinsically disordered. Protein, they don't have a structure at time zero as a monomer. They try to take up a shape because it needs to interact with its neighborhood. So once it interacts with the neighborhood, it forms a nucleus, and that's where it starts to pick up very, very fast, the kinetics. So it starts to self-assemble to form these long needles, which are beta sheet structure typically. This is what we see in needle structures found in plaques in Alzheimer's brain and type 2 diabetic beta cells and so on and so forth. Typically, one monitors at low resolution this kinetics of aggregation using a dye called thioflavin T, THT. Shown here is the thioflavin T intensity in the y-axis as a function of time. In real time, you can see it starts with the lag time, and in the sigmoidal fashion, you can see this aggregation happening, resulting in long fiber, needle-like structured fibers. In the lag time, you don't see fibers, and in the fully matured condition, you see lots of fibers. It looks like a two-step mechanism where you have no fibers and yes fibers, but monomer going to fibers, monomer in the beginning and fibers at the end. Interestingly, monomers at the beginning are good guys and fibers are benign in most of the cases. They're not toxic. So they look ugly, but they're not toxic. But in between are the treasure or most notorious species are formed. The monomer going to oligomer, oligomer converts into protofibers and then fully matured fibers. One can also look at using these microscopic images, including TEM and AFM. And of course, NMR is the best in my opinion, because monomers can very well characterize at high resolution using solution MR. And as the species becomes bigger and bigger, we can apply solid state NMR techniques. So my interest has been looking at the species that are formed in the lag time. That is right here in this dashed rectangular box in the lag time. It's not only monomers. Monomers can coexist with small oligomers. Small oligomers can convert into large oligomers. They can go into heterogeneous oligomers. They could be disordered. They could be structured, differently structured. It's really highly challenging to monitor this aggregation at high resolution. Almost all techniques fail because we cannot do crystallography, we cannot do cryo-EM to look at these dynamic events. But NMR, in my opinion, is one of the uniquely positioned technique can provide atomic resolution details. So these are toxics, therefore interest is huge here in characterizing high resolution insights in the formation of this toxic species, as well as their conversion into benign fully matured fibers. If we know the structure, we may be able to interfere these formation of these toxic structures with the compounds and potentially would aid in the development of drugs. So that's a goal. So shown here is a simple kinetic model one can think about. A monomer shown here in the leftmost green species slowly aggregates to form oligomer and maybe a nucleus. It's just a cartoon, so take it easy. Once the nucleus is formed, it quickly converts to protofibers and fiber. This process, fiber formation, is very, very quick. Typically, this nucleus formation is extremely slow and rate determining step in this kinetics. So we can do THT at low resolution. We can do TEM, which can give false information sometimes. But NMR, so let me tell you that I would like to focus only on IAPP. I might talk about a little bit here and there amyloid beta. So today talk is only on amylin or islet amyloid polypeptide, which is a hormone co-expressed along with another hormone called insulin. We are all familiar with insulin. Insulin is a hormone, controls the blood sugar level. And only in the diabetic case on the right side, you see the green spot coming from the fluorescence coming from IAPP aggregates start to kill this IAPP producing islet cells. On one hand, we need insulin to control IAPP and insulin to control the glucose level. On the other, IAPP start to aggregate and kill the islet cells. So it's like a spiral death. So here are some sample NMR spectra, 2D HMQZ, which can be optimized to acquire in few minutes, maybe now a fraction of a minute is possible as well if you have a friendly sample. At time 0 here, shown here at 25 degrees Celsius. So these processes are very sensitive to temperature and pH. So we reduce the pH. At low pH, this peptide doesn't aggregate. Interestingly, inside the islet cell, acidic pH is present. So that's one reason. That could be one reason why this peptide is non-toxic in non-diabetic patients, non-diabetic cases. So when you increase the pH, you can see sudden collapse in the disappearance of peaks here. What you saw, all peaks now, only few, and then completely gone into zero within 2 hours or so. So if we can monitor intensity as a function of time, monomer depletion is happening here. This is just similar to the THT-based sigmoidal curve. So this is sigmoidal depletion of monomer population, which is vanishing from solution NMR detection limit. One can also look at using solid state NMR spectroscopy as the species become larger and larger, or look at the fibers as well. There are a lot of great scientists working on this amyloid problems in India. I cannot say all the names. Madhu is one of them. Vipin is working on it. Anirban, and then you have Samir and Samret. And also you have Sudhita, lots and lots of people are working on fantastic amyloid-related problems. So it's possible that the fact that we don't see any new peaks appearing in solution MR time scale, we can rule out that the small size oligomers are not forming, or they are forming very quickly. They are converted into large oligomers. That could be another scenario. To further dwell into this kinetics and formation of nucleus oligomers in this process, we started to look at temperature variation. Shown here is at 4 degrees Celsius, I think. Yeah, it's 4 degrees Celsius. Even after 14 hours, you don't see any loss of signal intensity, no sign of aggregation for this peptide because the temperature is low, kinetic energy is low, so it doesn't aggregate. We also looked at using fluorinated peptides selectively labeled with the F19. F19 is really a very, very useful nucleus. Because high sensitivity, high gamma. So we can look at very quickly using 1D NMR in a fraction of a second. We can collect lots and lots of data. Shown here are the spectra as a function of time. We can plot the intensity and see the kinetics. As you increase the temperature, going from blue to green, the kinetics of aggregation is really fast. So suffice to say that with using any possible combination of techniques here, we don't see any intermediates here, oligomeric intermediates. So then we turned on to NMR again. We said, okay, let's look at Dorsey experiment. Fortunately, we took 2 different species, human and rat peptide. They're all 37 residues, very easy system to look at when it's monomer, very difficult system to study when it aggregates. Rat peptide, interestingly, doesn't aggregate. It's not toxic, doesn't form fibers. If you look at below 0, you can see here, only folded proteins would provide signal here below 0 ppm. So human peptide shows signal here. Also, there are several signals inside here which are not well resolved. We think that that's coming from well-folded oligomeric species, whereas you don't see that for rat. So we started to measure the diffusion rate using Dorsey experiment. We concluded that large oligomers are present, size on the order of 50 nanometer diameter. We also probed in different, with the different sample preparation. We can also see here, IAPP can form oligomers in a concentration dependent manner, so-called micelles-like oligomers, because there's a critical concentration above which it forms oligomers, below which it doesn't form. It's about 12.5 or 12-ish, 12 degree here, critical temperature, and the concentration varies whether it's C-terminus amidated or non-amidated. And also pH dependent, temperature dependent, and so on. So there is a critical temperature, critical pH, critical concentration for this oligomer formation. We can go forward and reverse to get monomer to micelles like oligomer and reverse. We also confirmed using AFM. You can see here above the critical temperature, you see lots of oligomers. Below the critical temperature, you don't see that. With the naked eye, you can see the oligomers in NMR spectroscopy. You don't see with the naked eye. People don't believe it, but it's true. It's micelles-like behavior. It's exhibited by IAPP peptide. How relevant are these micelles-like oligomers? So we can take 3 different scenarios. We can take the C-terminus amidated, which is biologically relevant, biologically occurring. So this is what is aggregating in type 2 diabetic case. The concentration is much, much lower. It's around 2, less than 2 micromolar. Whereas if you non-amidated C-terminus, it goes about 10 micromolar. If you take other peptides like rat peptide or 1 to 19 N-terminus segment of human peptide, they don't aggregate, they don't form fibers in our hands in certain condition. You can always make it form fibers, that's different. And also below this critical pH, they all don't exhibit micelles formation. So suffice to say that the micelles formation and oligomer formation is directly correlated with its amyloidogenic propensity for the peptide. This is also true for amyloid beta as well. So let me summarize based on saturation transfer experiments. Here are shown here 2D experiments at time 0 at the topmost row. And then if we can increase the pH, I think, yeah, pH 4, it doesn't aggregate. pH 7.3 above critical pH. You can see the aggregation happens at the N-terminus. So N-terminus come together, they start to self-associate to form oligomers. And then if you increase the concentration or temperature further, or concentration, the contact sites shift towards the core of the peptide, which is the amyloidogenic core of the peptide, which is about 20 to 27 residues region. This is also confirmed with IR spectroscopy, 2D IR spectroscopy, where they use very high concentration, much above critical concentration of IAPP. It was also confirmed for below below the concentration, but below the critical pH and below the critical temperature, above the critical concentration. Roland Winter found that association is at the N terminus. So our data is consistent with these two independent groups publications. We can also do other experiments. Here shown here is a sample slide on the amyloid beta, which is implicated in Alzheimer's. What you see here is we prepared a mixture of oligomer and fibers. You can see the THT binding to the fibers. You see nice intensity here. If you remove fibers, you don't see the THT signal for monomers and oligomers. So as in TEM, you don't see any fibers or oligomers in TEM picture, but you see in the mixed sample, you see fibers. In AFM, you see this filtered sample shows oligomeric species here. We can also look at that height here to understand the particle size. Now, if we take this solution dispersion of oligomers along with the monomers, we do HSQZ or typical NMR experiment, HMQZ, HSQZ, TROZ, NOZ. You don't see the peaks here because they're large in size. They don't tumble fast enough. So they relax faster. Our lines are broader. So as here is the monomers appearing nicely here, but the oligomers don't appear. Oligomers are in blue, the very few peaks, but pretty much there's nothing there. But so we do both solids and solution. We take this nano dispersion of oligomers in solution, we stick it at the magic angle, just spin the sample and look at using proton-proton NMR or just proton simple 1D spectra, we get beautiful spectra. Resolution is enhanced because you can remove the dipole-dipole interaction and any residual chemical shift anisotropy as well. So here's a sample experiment here. We did NOC and magic angle sample spinning. We can also do RFDR. We can spin out, suppress completely the proton-proton dipolar interaction, which is really small because this piece is still tumbling sufficiently fast enough. So the dipolar interactions are averaged. So we have only residual dipolar coupling. It can be averaged out even with 2.7 kilohertz spinning to a large extent, and we can recouple them. So the spectra are much better if you go up to 15 kilohertz. So we tried 10 kilohertz and 15 kilohertz. We cannot go beyond that for technical reasons. You can see here a beautiful NOC-like spectrum. We can do both NOC and RFDR. We see more cross peaks from RFDR because you can go long range correlation as well. So it's possible to quantify and do solution NMR like structure determination here using this simple RFDR recoupling technique. I'm also looking forward to using Vipin's latest RFDR technique and ultra fast NMR experiment. That is something we do for crystalline solids. I also would like to stabilize this oligomer and go into fast spinning. Maybe we can do distance measurement using this technique developed by Vipin Agarwal. So this is in collaboration with Bruker. We can also do other trick where aggregation doesn't happen in real time for NMR experiment to observe intermediates, but we can always make the sample to aggregate faster. Shown here is an example. I won't talk about this, but it's published here in ChemCom. We can stick the sample solution in the magic angle sample spinning rotor. Depending on the sample condition, you see this accelerated aggregation of this peptide, or you don't see any aggregation at all. If you have ultra pure monomers, you don't see aggregation. Most of the time, you don't get 100% pure sample. These peptides are extremely difficult to have with high purity, including the biologically produced one. You always have some impurities. So we see this aggregation happening here because you have at least very small, undetectable amount of aggregates, oligomeric aggregates. So what happens when you spin the sample, depending on the size of the oligomer, depending on the amount of oligomers or impurities, you could have different effect of spinning, physical effect on that collision between different species that would promote the aggregation go faster. This is our hypothesis because no way to observe other than just NMR experiments. One thing we did is to just deliberately contaminate the sample with the EGCG green tea extract polyphenolic compound. So now we have two species. One is organic or polyphenolic compound and peptide. They can collide. We can see the pi-pi interaction triggering this peptide aggregation dramatically. So I would like to change the topic here and then focus on membrane-centric biological questions here. Amyloid polyprotein, polypeptide protein, or amyloid precursor protein, APP. This is the protein where enzymes come and cleave at this cleave site, see where it's shown by scissors. So then it generates the amyloid beta peptide. The fact that this peptide is generated in the membrane environment, it's exposed to the lipid bilayer immediately. So lipid peptide interaction has to play a role, right? This peptide is also present in serum. I'm sure there are free lipids as well. That's something we're probing. So my interest has been looking at this peptide aggregation in the membrane environment by interacting the peptide with the lipid bilayer. The scenario becomes extremely complex. A monomer can aggregate in solution phase to form fibers. A monomer can quickly pull towards the lipid membrane bilayer. It can self-assemble to form pores and disrupting the membrane, or aggregate to form fibers in the process of formation of fibers that can disrupt the cell membrane structure that's implicated in the toxicity induced by this peptide. If I have to look at high resolution details of all these structures, it would be great for the drug development and to fully understand the real, the biological, biochemical processes that's happening in my brain and Alzheimer's patient's brain. That would be great, but super, super challenging. Because the factors that control this aggregation kinetics and the species formation and their heterogeneity and their lifetime is so many parameters. The lipid type, lipid composition, curvature of the membrane, and then the temperature, pH, lots and lots of parameters, and peptide local concentration and so on. But this is where a great opportunity for tool developers, including NMR experiments. We also develop some biophysical approaches and chemical tools as well. You can chemically control, using small molecules, you can control this aggregation pathways. So these aggregation do disturb the membrane. Shown here is the data. Even the lag time, you see significant dye leakage coming out by disrupting the membrane. So suffice to say that the two different mechanisms, fiber independent step one, fiber dependent step two. I will quickly go through this slide. So fiber dependent ones can be probed using static aligned samples in aluminum oxide nanodisk, where you take this lipid bilayer, get it sucked into this aluminum nanodisk and monitor P31 NMR. At time 0, you see here this aligned peak. At time X, the peptides self-assemble to pull out the lipids to form isotropic species that tumble fast enough to give this isotropic chemical shift peak. So it's fragmenting the lipid bilayer, causing cell death. We also solved this intermediate structure using detergent micelles. They are not good model membranes, I hate it, but they are good tools to trap the intermediates, because these intermediates are heterogeneous. They're quickly changing in their status. So we can use micelles to trap these intermediates and solve the structure using traditional NMR spectroscopy. So we solved the structure of human peptide and rat peptide is disordered here. There are prolines and so on and so forth. I won't go through the details, but my interest in trapping this intermediate in planar lipid bilayer, which is more biologically relevant. Thanks to Steve Sliger and Gerhard Wagner, Gerhard Wagner gave us a real jump start in this arena of a nanodisk, which is like sushi. We have this belt. Nori is the belt, and you have this planar lipid bilayer. Rice is like lipids, and you have meat is protein. So all these are happening. So we use it for membrane protein redox partner complex formation in this nanodisk. We also use it to trap this intermediates. So I also need to thank my colleagues in Munich. Thanks to the Hans Fischer Fellowship. Thanks to my host, Ben Reif, and my colleagues, Ben Reif and Franz Hagen. With their collaboration, we started to look at the condition where we can trap this amyloid intermediates for IAPP peptide in nanodisk. Shown here is the empty nanodisk with the TEM picture. It's beautiful. You see the small dots. They're low resolution though. And if you aggregate the peptide in solution, you get these beautiful fibers. if you aggregate the peptide in cleverly chosen after screening several conditions, you have a small amount of anionic lipids with the major zwitterionic lipids, we're able to trap this intermediate where we don't see the fiber. If you zoom in, you don't see much, but one can always overinterpret that. Nanodisk may be distorted. Okay, that's overinterpretation. But we can go ahead and look at the peptide by labeling it differently. Here's the nitrogen 15 label. You can do HMQZ, HSQZ, TROZ, and 3D spectra. We can also trap the monomer and see the monomer, how it interacts with the nanodisk as a function of time. We think that based on this experiment, the N-terminus gets sucked into the core of the bilayer, similar to what we have seen in this detergent micelles. Okay, so it's a helical domain. It's hydrophobic enough to go inside the greasy layer of the membrane. But we can also trap oligomer, IAPP oligomer, and we can use carbon-13 nitrogen 15 labeled peptide, do the 3D NMR, assign the peaks, get the chemical shifts, and do the Rosetta, and build a structural model, and then do the PRE to just see where this oligomer is located in the planar lipid bilayer, including the doxyl labeled lipids, and use shake and bake empty simulation approach. We get this high resolution structural model, I would put it. The model looks like this. You got 3 anti-parallel beta strands, and the loop region here is the core of the amyloidogenic core of the peptide. NF Gale is the most amyloidogenic core. If you take this peptide alone, it would aggregate quickly to form fibers. So in the final fibers, this is in beta sheet structure, but the fact that this is in disordered turn region suggests that this has to be a oligomer, and we don't see any fiber in TEM as well. So this has to be a small size oligomer, maybe 4 to 8-ish monomers. This is 1 unit of that because we're able to digest using solution of mass spectroscopy. So this is the first ever oligomeric structure at high resolution. So you can also use high resolution or ultra fast, high speed AFM. You can see the fiber growth in real time here for IAPP. And if you use nanodisk, this is synthetic polymer nanodisk, which we developed in our laboratory here by Thiru. You see there's no aggregation happening. Okay, so left and right here with the nanodisk is the same here. You can also see the publication we published, I forgot somewhere, I think JMB or ChemSci. This is done by my postdoc, Bikar Sagu, in collaboration with Nakayama and Andozan. So you can also see here, this nanodisk alone here, they don't aggregate. IAPP does aggregate to form fibers in the central one, and then in the nanodisk, it doesn't aggregate. So the fact that we're developing polymer nanodisk, we can also align them. We can form large size nanodisk. They have susceptibility. They can align the magnetic field, which means that we can do lots and lots of cutting edge NMR experiment, including residual dipolar coupling, residual, any anisotropic interaction can be measured. Here is an example of cytochrome C. Cytochrome C doesn't interact with the zwitterionic membrane. So it has to be in the solution phase of the nanodisk. We can measure the RDC and correlate with the determined, theoretically predicted one. And we can do that for different structures, and we can approve which structure is correct or which structure is incorrect. So the RDC depends on the concentration of the nanodisk as well. So 40 milligram is this much, and then if you have 100 milligram per ml, you can have larger dipolar coupling, resolution can go down as well. We can also use RQC quadrupole coupling. Obviously, many people use the deuterium NMR, but we could do O17. The initial part of this work, Ganapati was involved he came to our lab from India as a visiting scholar, and he gave us a jump start in the beginning days, and then later on, my postdoc Thiru and Bikash continued and then expanded his work to the nanodisk. Lots of people have contributed to this O17 NMR, Gang Wu, Woolfield Gang Wu, Griffin, and Malcolm Levitt published a fantastic paper on trapping water molecule in liquid crystalline matrix, and Buck Ball, and fully characterized all the 5 transitions. And that's what we see here, center transition and then satellite transition, 5 transition, because the 5 half nucleus, there are 6 energy levels, 5 transition, we can measure that. I won't go through the details, but we can measure RQC. RQC depends on the exchange between free water and bilayer bound water. that depends on the extent of alignment. If we go to higher field, we should have higher RQC. And we can measure T1 values for this transition. They're all pretty similar within the experimental errors, but they're fast. We can also do the bilayer normal perpendicular to the magnetic field. We can use paramagnetic solve to change the susceptibility to flip it by 90 degree, and the parameters or the interaction or tensors switch by 90 degree via the second order polynomial 3 cosine squared theta minus 1, nitrogen 14 quadruple coupling doubles, P31 shifts by 90 degree in the frequency space over 17. Doesn't double because water molecule is not in the lipid matrix. It's really transiently associating. So this is for low concentration of the lipid, this high concentration of the lipid. So I think my time is running out. So we also use this lipid-centric aggregation to screen compounds. We identified 5 excellent hits. We are working on using high resolution NMR experiment to look at where exactly they bind, how we can develop further to interfere with this aggregation using these molecules as chemical tools. So with that, I would like to acknowledge all the people who worked for me. This is all their work, and I'm here just to present their work. Bikash for his fantastic contribution on amyloid aggregation, nanodisk, and polymers, and Thiru for the development of synthetic polymer. Thiru is coming from Athreya's lab. Krishna is coming from Athreya's lab. Krishna worked on A-beta interaction with the different proteins. And Sarah Cox involved in this drug screening and development, and others as well. I would like to thank all my collaborators and funding, and thank you all very much for your attention. I'll be happy to answer your questions.
Well, the topic is open for questions. We would like to take about 5 to 6 minutes for answering questions.
Sure.
Yeah, any questions, please raise your hands or mention your name and use your microphone.
So there is a question from Jeet. Jeet, you can ask a question.
Go ahead.
So my question is, when you do the pH titration and see the disappearance, regular disappearance of peaks as a function of change in pH, I see that the peaks which are not disappearing do not change their chemical shift positions, right?
Yes, this pH is not dramatic change, so we made sure that,
okay.
I don't remember, so that's one is sample spectra that I've shown. You can look at our publication, I think.
Went from, I think, 5 to 8 or something.
Correct. We made sure the peptide is not degrading. Yes, chemical shift can change. We are going to apply the correction for that, and with that, we determine the change due to the peptide oligomer formation. We also measured independent using other experiments like pyrene fluorescence assay and AFM and other experiments as well.
Okay.
Vipin has a question.
Yep.
Vipin.
Hello. Am I audible?
Hi,
Yeah, you're audible. Yeah.
Okay. So very nice talk. I just had a question regarding your, somehow the oligomers don't seem to evolve with time, even when you are doing a mechanical rotation. So you would need energy, and this energy could be provided technically by mechanical rotation for oligomers to convert into higher species. But in all your experiments, that is not happening. Why is that?
Well, this is an excellent question. So it depends on the type of oligomers. If you look at our ChemCom paper looking at the monomer going to oligomer, that does evolve to form fibers, right? Due to mechanical, and so agitation, your mechanical rotation can induce different effects on different species that does accelerate the aggregation. But on the A-beta, what we did with RFDR, it does evolve in size. We see that not using NMR, we can see it clearly using, if you stop spinning, take it out and look at using DLS, if you believe in DLS, dynamic light scattering, we see the particle sizes changing. And, but we are spinning slow enough that we don't see the contribution from fibers.
So you actually never form fibers, is that correct?
Yeah, we don't see the signal from fibers. Maybe there are only mobile parts are contributing to the spectra, but by and large, we only see the signal from majority population, which are oligomers. And proton-proton NMR doesn't take that much time, it's very quick.
You also had the three-dimensional carbon nitrogen spectra. So on,
Well, those are
I
oligomers
think
you are talking about. Those are oligomers
yes.
trapped in nanodisk, which is solution
Okay.
NMR, not magic
Okay.
angle spinning. Solution NMR.
Okay.
We did apply NUS. We tried to do that. It was very difficult. Sensitivity was very poor. It was challenging, quite challenging as well.
Okay. Can I ask one more question?
Yeah.
So you showed the sequence where.
By the way, let me just correct back. The oligomers, what we solved, characterized using RFDR is off pathway oligomers. That's another reason why they don't quickly form fibers. They're not.
So they don't evolve with time.
They don't evolve to form fibers in our hands. So we can always make it to go to form fibers, but even after 7 days, they don't go to form fibers. So
okay,
these are good oligomers that can be used for ultra fast spinning as well.
Okay.
Yeah. Sorry. Go ahead. Yeah.
Yeah.
We'll okay. Okay. Go ahead. Make it quick. Yeah.
You had shown the sequence where you did the proton-proton RFDR on the oligomers. And the sequence doesn't have anything special for water suppression. Why do you get such a clean spectra?
That's a very good question. We did use a combination of D2O alone and D2O water. And we did play with several different things. And we also played with the amount of water as well. Maybe we did water suppression. The pulse sequence doesn't show.
No, it doesn't show. I was very surprised.
The simple pulse sequence just to show the noisy in RFDR. I go back and look at the program that we use. This is done, 15 kilohertz spectra are carried out in Bruker. So we did use water suppression, yes. Yeah, okay. Water suppression is an issue, but we did use it, and we did use D2 as well.
Okay, thank you.
There is a question from Neeraj. He's asking, can you comment about rotational diffusion of nanodisc during mass?
Can you repeat that question again, please?
Can you comment about the rotational diffusion of nanodisc during your magic angle spinning experiments?
Oh, we did not use nanodisc for magic ensemble spinning yet. We are done for bicells, so we expect that to be similar, but that could interfere with the type of experiment that you would like to do, because the time scale does matter. They can interfere. You have to cleverly not constrictively to get high resolution spectra. So that could be an issue. You need to optimize the conditions. So we are not done that yet.
Okay, and there's one quick question from Dr. Jagadish. He's asking, can you explain what is the effect of mutation on IAPP?
Oh, hello, sir.
Yes, go ahead.
Good evening, sir. It was a scintillating talk. Just my question was what is the effect of mutations on IAPP? Since we know that some of the mutations will slow the aggregation and some, foster the aggregation. Have you worked on that, sir?
So a lot of people have worked on the mutations, because the fact that you have different species have different IAPP, some of them don't aggregate, some of them are not toxic, some other species don't develop type 2 diabetes. So we have compared rat and human. Human has histidine at 18th, H for histidine, and then rat has arginine, R. We played around with this. So if you mutate histidine, that has significant effect on the kinetics of aggregation, as well as the type of oligomers you form and so on. Martin Zanai in Wisconsin has done fantastic work on this in this area. Roland Winter has done nice work. But in our hands, a rat peptide doesn't aggregate, doesn't form fibers. We see this helical structure very similar to human IAPP in the N-terminus. And we see very different behavior for membrane interaction, in addition to the aggregation property. So I hope that clarifies your question.
Yes, thanks a lot.
I think with that we will
there is
now. If there are more questions, it can be sent to us, and we will send it to Ramamurthy for answer by email later on. So let us thank Ramamurthy for a wonderful presentation. Although the time was limited, he has done a wonderful job. Thank you, Ramamurthy.
Thank you very much. Thank you very much.
Yeah. The next speaker.
You can stop sharing your screen, sir, and then Professor NSP.
Was there a question or interruption? I couldn't hear it.
No, sir, you go ahead.
Okay, all right. The next speaker, the second speaker of this webinar series is Professor N. Surya Prakash, fondly addressed as NSP. He is currently Professor and Chairman of the NMR Research Center, NRC, at Bangalore, and also associate faculty of the Solid State and Structural Chemistry Unit at IISc Bangalore. He was the immediate past president of the National Magnetic Resonance Society and helps with the functioning of the Bangalore NMR's office immensely. So personally, I thank him for all the efforts he puts in to get the NMRS running. The major areas of research of NSP are on the development and application of NMR spectroscopic techniques for discrimination of enantiomers, study of weak molecular interactions, and development of multidimensional methodologies to simplify the analysis of complex NMR spectra of scalar and dipolar coupled spins. We have developed several sequences to address the challenges involved in the analysis of spectra. So, Professor NSP, you have the next half an hour for you.
Okay. Thank you, Professor Subramaniam, for the nice introduction. In fact, I'm going to, it is a big change from biomolecule to a small molecule now. I'm going to talk something about our recent work and not recent, couple of years back, work on clean sequences, especially for the extraction of coupling for one of the selected protein in a complex molecule. And it has a lot of artifacts. How do you completely eradicate these axial peaks, etc. That's what I'm going to talk today. But before that, I would like to dedicate this talk to my colleague, Professor H.S. Athreya. And he's really emotional, and it was really a very, very premature departure of him. I'm really shocked. I was wondering whether I will be able to give a talk today or not. With all this that happened in the last about 2 weeks, and a sudden change, and I was monitoring the development, what is going on when he was admitted, right from the day he was admitted to the hospital till the last day. And every day I was getting the reports and I was losing hope. And he was the one who initiated this type of activity for webinar series. And in his absence, I was also feeling nervous what to do like that. But anyways, I don't know what to say. He's a very close friend of mine, very close colleague. He's more like a younger brother for me. And we were all the time together. Right from the day when he joined NMR Center, till the day he orphaned us, he was with me. And we used to discuss varieties of subjects, be it academic, non-academic, property dealing, share dealing, whatever it is at the coffee table. It was a fantastic discussion we used to have. Nice human being, always with a smiling face, ever smiling face, bubbling enthusiasm, and never used to hurt anybody by any of his statements. Fantastic for human being. So with a heavy heart, I would say we are unhappy that he's not with us, although he initiated this activity. So I always start the day assuming that my friend is still sitting next to my office. The day Monday when I entered the office, I was really taken aback. I had to pass through his chamber. It was so difficult for me to walk into my office because that was haunting me. But I still assume that he's with us and he's sitting next to my office, and I continue to start the day. And then, of course, before we start, one more thing I wanted to say is, when this thing happened suddenly, I was getting nervous. Last 15, 20 days, I was concentrating on other things about his hospitalization and what to do and physical help extending for his family. Then what about the webinar series which we initiated? And it was all, he had all the information, data collected, everything. I really was nervous. Then I would like to take this opportunity to thank Jeet and Vivek, both of them, really bailed me out of the situation. They did enormous amount of work to keep this going, and they have brought it to this shape today. Jeet and Vivek, you are there. I take this opportunity to sincerely thank you for all your efforts. You are with me all the time. And I am sure you are Athreya 2 and Athreya 3 for me. So with this, let me continue with my presentation. And I would say, first I want to thank my contributors of this work. And of course, there are many of my students, a lot of people working in different areas. And the work that I'm presenting today is by 3 students. One is Neeru, Lokesh, and Sandeep. These people contributed for this work. Of course, without money, nothing can be done. And funding was by SCRB, New Delhi. And of course, let me start with the spectral complexity. I'll take the example of the weakly coupled spins. When I take the example of weakly coupled spins, sometimes not only biomolecules, even small molecules is so complex. What we are interested, especially chemists or pharmaceutical scientists, want to get the spectral information like chemical shift coupling, constant dipolar couplings, etc. Sometimes we get into serious problems. Let's start with this. start with a simple molecule. I'm concentrating on proton A. And of course, simple analysis tells me the pattern has to be a septet. Of course, we can get the coupling information and chemical shift in this small molecule. Go to the next molecule. These are all hypothetical molecules which I have designed just to written the structure just to explain. Now look at this proton in this molecule, which is going to be triplet of a quadrite. No problem, we can analyze and get the information. Go further, keep on increasing the complexity of the molecule. As you go to the next molecule, we are going to get quartet of a quadrite. The real problem comes if you go to the molecule like this. And it is a simple proton coupled to 3 different chemically non-equivalent protons. And we are going to get a pattern like this, which a stick plot in which I will say it is called triplet of a quadrite. Okay, with some difficulty, we can understand and try to get its spectral information. Now, these are all 4 hypothetical situations where I am explaining to you by stick plot. What happens if I go to a realistic situation like this? I am looking at the molecule, these are called fluorine substituted benzene, which is some of the molecule designed and synthesized by our collaborator for X-ray studies, and then we took it for NMR studies. Now in this molecule, when we are trying to understand weak molecular interactions, we are looking at a fluorine NMR. What happened to this molecule when you look at the fluorine detection, look at it, and there is no interaction between the spins of this phenyl ring and spins of this phenyl ring. And of course, there are 4 protons and a fluorine and also NH proton. Incidentally, this NH proton is also coupled to all the spins of this ring. And this NH proton is also coupled to all the protons are full enough this phenyl ring. And this is a weakly coupled spin system. In NMR jargon, as you all know, we call it AMPXY spin system. And since we are looking at the fluorine, and the 2 fluorines are chemically inequivalent, if I look at the fluorine 5 and fluorine 6, a simple calculation tells me I must get 32 peaks for each. Of course, in this fluorine 5, I'm seeing only 16 peaks. One of the coupling is negligibly small or 0. We will not worry about that. But look at the fluorine 6. This is a pattern we are going to get. It's a multiplicity pattern. In fact, I must get 32 peaks. How do you resolve that? There is severe overlap because lots of couplings are present. This fluorine is experiencing coupling with 4 protons and also NH proton here. So in such situation, what do we do? This is where, this I gave a real example. And our efforts in our group is always to address few challenges in the analysis of the complex spectra of small molecules, be it scalar coupled or dipolar coupled. And our direction of approach always is to see how do we get a high resolution, how do you enhance the sensitivity, speed up the experiment, and discerning of the degenerate transitions, unraveling of the spectral overlap, and simplification of the spectral complexity, and facile extraction of spectral parameters. These are some of the examples which we are trying to do in our group. But today I'm going to tell you about number of sequences that has been designed for the information retrieval in the proton NMR spectra of small molecules. And these are some of the examples of our designed pulse sequences, which we have given different names like Zoltoxy or various varieties of names which we are given here. And today I am going to deal with what are called clean sequences. And the clean sequences in that what we are trying to do is, first of all, this one, it is called clean G-serve. Second one, it is called pure shift edited clean G-serve. And other one is called clean perfect echo serve. These are the sequences which we call as clean sequences. I'm going to discuss how we derive the spectral information using these sequences and what are the challenges we try to address this. Of course, first of all, as I said, most important thing which we have to do, our main aim is when we are analyzing small molecule for whether it is for the chiral discrimination or for the weak molecular interaction study, we need to extract the coupling information or analyze the spectrum to get the chemical shift information. First of all, the extraction of the coupling information for a complex multiplicity pattern is a challenging task. Of course, it is well known from the J-result experiment in the early days of NMR, 2-dimensional NMR. It is a powerful experiment which was published by Professor Hunt, like a spherical sequence we can use to get the J coupling. And in this experiment, as all of you know, each proton gives full multiplicity in the information in the F1 dimension. And of course, in the direct dimension, we have J plus chemical sheet both present. And when I do the 45-degree tilting, this is what I'm going to get, where I have supposed to get the J coupling here and chemical shift here. But look at this example of a molecule like this. I'm concentrating on the proton, which is highlighted here. This proton, incidentally, experiences coupling with number of other protons present here nearby, which are scalar coupled. And look at the complexity here. In such type of system, is a simple molecule with many couplings, severe overlap, and because of the poor resolution, we will not be able to get the information what we want. So then a development took place in the 1995. The Fake and Berger group proposed what is called the selective refocusing experiment, where you can extract a single coupling between any two protons coupled, for example, A and X, two protons coupled to each other, we can extract the coupling. But it is similar like your J-resolved experiment. Only thing is in the first one, you are going to apply on a particular proton in which you are interested to extract the coupling with another proton, apply on this proton a selective 90-degree pulse. And then you apply 180-degree pulse selectively on the protons E and X together, and similar to your J-resolved experiment here. And what happens in the sequence, without going more to the details, you will see that in the F1 dimension, we get the AX coupling between the two protons, one which is we wanted to determine, and another proton in which you applied both 180-degree pulses. And what happened to the remaining protons, proton couplings? All the other couplings are suppressed. So we get only one coupling of one proton connected to other one, its coupled partner. Now let us see what type of complexity we are going to come across. Look at this molecule. When a proton experiences many couplings, I have taken an example like this in this proton 15E. This is coupled to, incidentally, 4 other protons, scalar coupled. If I have to follow the selective refocusing experiment, which is published earlier, what you have to do is you have to do 4 experiments. Each time, because we are going to get only one coupling of proton 15E with other coupled partners. So 4 couplings, we have to do 4 experiments. In simple terms, we need to do n experiments to get couplings, required n coupling information for the selected proton. But it means the selective refocusing experiment is very time consuming. That's one part. Okay, that's apart from that, if I have to do, instead of avoiding doing multiple experiments, can I do the similar experiment in a first it's possible way? Instead of doing n experiment, can I do one experiment, get all the scalar coupling information exactly using the SURF technique? Okay, it is possible. We all know we can do the selective excitation by applying a soft pulse and selectively excite any one of the protons of the spin system. What happens if I have the gradient encoding? This is also well known. When you do that, because of that gradient application, you can select individual protons in different slices. In this, for example, in the molecule given here, we can have proton 1 excited in this slice. As you keep on increasing the gradient at different slices, you are going to excite different protons and we are going to detect them. What it means is, it is like a selective excitation of different protons in a single experiment instead of multiple experiments. So this selective technique, it means if we adapt that for the selective refocusing experiment, it is possible. So only difference is we have applied only gradient here. So this is what is called the gradient selective refocusing experiment. It's a blend of selective excitation with selective refocusing. And we are going to call this as, it's not me, it is in the literature, it is called as gradient encoded selective refocusing experiment. Fine. So what is the advantage of GSURF over SCRF? Just now I explained to you. Of course, this is the SURF sequence. It's slightly different from what I showed earlier because modifications will keep happening in the literature. But in the case of SCRF, you can see, I look at the proton E, which is coupled to 4 different protons, which are scalar coupled. Now, I can get 4 coupling in 4 experiments. For example, in this experiment, if I do, I can get only coupling between proton E and B. Then what about others? Now, for others are not seen here, and I have to do 3 more experiments. Now, on the other hand, just by the application of this gradient encoding, we call it is GSURF. Now, this advantage is this proton E, which is coupled to the remaining 4 protons, all the couplings are seen at a time. This is the fastest way we are going to get coupling. It's a beautiful experiment, but there are weaknesses for it. It's a 2-dimensional experiment. We can get multiple coupling in a single shot, but So some weaknesses, some issues are there related to this gradient encoded selective refocusing. What is that? This is a simple molecule called strychnine. I will show what happens if I take the G-surf spectrum for this molecule. Okay, now let us concentrate on proton 15B. The proton 15B is coupled to 3 chemically inequivalent protons, 15A, 14, and 16, which are highlighted here. Okay, now this is the G-surf spectrum. Immediately you can see lots of peaks here. What we should expect when I say G-surf experiment should give you 3 coupling simultaneously which are coupled to proton, then I must get only 3 sets of doublets in the indirect dimension. But look at it, in addition to 3 sets of doublets which I'm expecting, we are getting strong axial peaks at 0 frequency, which is coming from uncoupled protons. At the same time, you see the evolution of unwanted couplings are also present. These are things which are not coupled to this proton 15B, but you see this can mislead you. It will, we are not going to identify the true coupled partners because of this. These are some of the weaknesses of G-surf experiment. So what we have to do? What we thought is, can we clean up the sequence? Can we overcome this weakness? So this is the G-surf sequence which is well known. And what we did is we appended a block like this, small block, which contains a selective 90-degree pulse, followed by a delay and a hard 90-degree pulse. Of course, other things are there, which small modifications which is shown here. This is nothing but the G-surf sequence. Only for the G-surf sequence, we appended small block here, pulse sequence. Let us see what is the role of this initial pulse block. Without going more into the details, you can see after the application of 90-degree pulse on a selected proton, let us say proton A in this case, you can go through that from IZ, you will bring it to IY, allow it to evolve for some time delta during the duration. And these are the couplings which evolve, which all of you know, I don't need to go through the step by step. And then after second 90-degree pulse, these are the terms which comes. But interestingly, what is going to happen is we will remove this term. This will not be there. And this term, we are going to take it and feed it back to G-surf sequence block. This will be the appended part for the G-surf in the initial appended part. And this part will be fed into that. And this, what is the effect of this additional pulse block in the sequence? And without going into the more details, I'll show you the spectrum and what is going to happen in the sequence, how we can overcome some of these difficulties. This sequence removes the presence of huge axial peak at 0 frequency, which is the basic intrinsic weakness of the G-surf sequence. It will remove that. Secondly, the appearance of the additional peaks from the evolution of unwanted couplings, which will cause confusion for you to identify in the identification of the true coupled partners of the selected spin, that also will be removed. That will also be overcome. So since it cleans up the spectrum like this, we call this as clean G-surf, where all unwanted artifacts are cleaned up in the spectrum, and it is again a G-surf experiment, and we are going to get a fantastic spectrum. So we'll before going to the experiment, as I said, this block is appended. We want to know what is this delta. Okay, so this is the delta which we need to know. What is its value? How do we define that? For that, it depends upon the strength of the individual coupling. If I have a selected proton, how many protons it is coupled to? It depends upon the strength, which anyway is not identical, which will be different for different protons with the different coupling strengths. So we need to experimentally optimize this. So of course, we will not be able to get for all the coupling one single value, but we have to get an optimum value which is good for utility. So what we have to do is we did an experiment like this. What we have to do is keep on varying the delay delta. How we do it? See the delta delay, of course, the time in the order of millisecond we have to take. Keep varying the delay and see what happened to the signal, how does it evolve. And our aim is to see that for a particular delay, the signal intensities, all of them should be maximum. Let us not worry about the phase here, that all will be taken care. But only thing is that we have to ensure that all the signal intensity should be of nearly equal intensity. Okay? You will not be able to get exactly, but nearly of equal intensity. And when you do that, you will find out for this value of delta, more or less all the signals which are of equal intensity. So we choose this as the value delta, which is optimized for a particular sample. This is one of the drawbacks, because if I have a different sample, we need to optimize delta again. So it is not that we optimize for one molecule and use it as a constant value for all our experiments. That is not possible, but we need to optimize experimentally for each sample. Okay, now we have optimized this, let us say. After optimizing, we applied this on a small molecule, a menthol molecule, where we consider the proton 6e. This is the proton. We have equatorial proton for 6, and we chose that for selective excitation. Of course, one of the reasons should be that it is well isolated. Otherwise, selective excitation of a particular proton in a crowded region like this is very difficult. It is very challenging. So we always choose something which is isolated. We took the isolated proton here, 6e, and since it is coupled to proton 6a, proton 5, and proton 1, we must expect 3 sets of doublets in the GSURF experiment. And we did the GSURF experiment for comparison study. This is a conventional GSURF experiment where you can see huge unwanted peaks. We are going to get huge lump of peaks here. And at the same time, these are all clumsy things you can see. And there's also strong axial peak, which is coming at 0 frequency. So this is the thing which is confusing if you want to get. As I said, it is coupled to 6A, 5, and 1. 6A is hidden here. So you cannot expect to get the accurate value of the J coupling here because it is in this mess, which is inherently, it is present because of this sequence. So now we did the comparison with our clean sequence after optimizing the delta value for this molecule. We will run this experiment. This is our clean GSURF experiment. As you can see clearly, it completely eradicates all the axial peaks and there is no evolution of unwanted coupling. It is a clean spectrum, clean GSURF, where 3 couplings for this proton 6e can easily be measured from this doublet separations. Very precisely we could get it. Okay, this is for a small molecule coupled to 3 remaining protons. Now what happens if we go to a slightly bigger molecule like strychnine, which I showed earlier is how GSURF worked. Again, I'm comparing this with the same molecule. Again, let us consider the same 15B protons. We are supposed to get a coupling of 15B with the remaining 4 protons, which I said earlier. And this you see, this unwanted evolution of it, extra evolution, which is not part of our coupled pattern. It's not this 15B may not be coupled, all those things. But when you see that, this is also strong 0 frequency axial peak. And when we do our clean GSURF experiment, you can see which is coupled to 16 and 14 and 15A. Clearly, 3 doublets we could get from in the indirect dimension. And no confusion, no artifact, no overlap of the unwanted signals. And very clearly, we could get coupling information for one of the coupled protons simultaneously in one simple experiment by gradient encoded selective refocusing, at the same time cleaning the pattern. Of course, the question is for most of the times chemists, when they want to do the synthesis, there will be a lot of mixtures analysis there. We have to see the mixture analysis. So we took a binary component mixture of 2 molecules to see how the spectrum will be there in GSURF and clean GSURF. And this is GSURF experiment for this molecule. We are concentrating on one of the proton here, proton 12, and then which is coupled to proton 13, 14, and 15. And look at this GSURF experiment. Certainly, you will see a lot more extra peaks are there, lot more unwanted signals. Lot more unwanted signals are also present in this. As a consequence, it is very difficult to identify the true coupled partners. Whereas our clean GSURF experiment not only removed all those unwanted signals, including axial peak, we are able to measure the coupling of the order of 1.2 Hertz, which is hidden here, which is not possible to get from the conventional spectrum. So clean GSURF, because it is able to clean this type of overlap of axial peak, we could measure very precisely coupling of the order of 1.2 Hertz. Okay, So of course we wanted to see, let us go to a very, very complex case of a very severe overlap. We took 3 different molecules where they can see the lots of overlap will be present here. And in this case, again, for a molecule like this, if you wanted to extract the coupling of proton 2 with the remaining protons, which is coupled to, you are very clearly we are able to extract the coupling for this molecule in a combinatorial mixture, whereas this type of messy things which was present, we could completely remove it. Okay, this is what is a clean G cell. Now the question is, when we try to append the pulse, another block of the pulse sequence, everything, the question of sensitivity came into the picture. And now we wanted to see what is the sensitivity of the G-surf experiment. I'm sorry, clean G-surf experiment over the G-surf experiment. We did some experiment at the same molecule under identical conditions. As much as possible, we kept it timings, everything same, except sometimes with the pulse sequences of the order of little bit of few microseconds cannot tally. But doesn't matter. But most of the majority of the experimental condition is kept constant. We did the comparison of the signal intensity. Of course, with the clean G-surf sequence, there is a sensitivity penalty of about 15 to 20%. But nevertheless, we are able to get a clean spectrum without any confusion. We can get coupling information. Okay. With this, we wanted to go for the next step. We wanted to take a blend of real-time homonuclear decoupling with the clean G-surf sequence. What will happen? This is what is called as pure shift-edited clean G-surf. This is, we call it as PS clean G-surf. Of course, pure shift experiment is well known. I don't have to explain to this audience, but it is a real-time manipulation of the free induction decay. When you are applying the free induction decay, what you have to do is you apply the J refocusing at different regular intervals. And then what you have to take is you have to take this data, which is present in the intermittent period of the J refocus. This intermittent FIDs are collected. We need about typically 16 to 32 or 64 blocks of such thing, depending upon how long the FID decays everything. And then this type of data is collected and concatenated all put together and do the Fourier transformation. Now, what the spectrum you are going to get is pure chemical shift and no chemical shift will be present. No coupling will be present in that. Only chemical shift information will be present. And this is, we wanted to try for the sequence. And this is what is the regular clean G-surf sequence, for which we, on the right side of it, we appended the real-time broadband homonuclear decoupling sequence. In which case, when we did this, what we should expect is in the direct dimension, there should be pure shift. That means chemical shift and J coupling, which are present, both present in the direct dimension, J coupling will be removed and only chemical shift will be present. And in the indirect dimension, as in the previous clean G-surf, we must be able to get coupling information. So we adapted this trick and the same molecule. Now, this is the clean G-surf experiment. As you can see, there is no artifact, no zero frequency axial freak, and no additional evolution. And this is the clean G-surf. When I expand each of this region, you see this is the direct dimension. We have J plus delta, and we have J in the indirect dimension. Now, spectral complexity is still when it's persisting in the direct dimension. We appended that homonuclear decoupling, and in this pure shift-edited clean G-surf, we removed the coupling in the direct dimension also. So very clearly in this experiment, what you are going to get is J coupling in this dimension, indirect dimension, and only chemical shift for each of the chemically inequivalent protons, which is coupled to the selected proton. And spectrum is very simple now, free from multiplicity pattern in this dimension, and it gets a good resolution. So of course, this is what the high resolution is imminent. And similar experiment was also published called push G-surf, which is again called pure shift edited G-surf, which is not a clean sequence again. It has some weaknesses. And this was published way back in 2015. And then we compared our PS clean G-surf with push G-surf. And of course, we have unwanted evolution also here in this case. And of course, zero frequency artifact is also present there. But in the pure shift edited clean G-surf, there is no such problem. And we are getting a very good spectrum. Again, when we made the comparative sensitivity comparison, incidentally, our pure shift edited clean G-surf has a better sensitivity here, of course. And we wanted to compare all the three experiments, push G-SERV, clean G-SERV, and pure shift edited clean G-SERV. These two are our sequence, this is a published sequence. And you can see that in this clean G-SERV, we have a J coupling and dipolar coupling both present here. And in this case, only J chemical shift and J coupling here. And in the push G-SERV, of course, the decoupling is there in the direct dimension, but the artifact is still present. And now there are two issues present in this, all these sequences. One, when you do this, sensitivity is one thing which we have to consider, because when you do the selective excitation, and there is a bandwidth issue problem. And then, of course, strength of the encoding also limits the thickness of the slices. It gives us what is called the penalty of the sensitivity. So next issue we have to address is to increase the sensitivity. I mean, at least avoid loss of the sensitivity as much as possible. And for this, very recently last year, a sequence was published by a Chinese group, which is called PE-SERV, which is nothing but perfect echo selective refocusing experiment, which circumvents sensitivity issues of G-SERV. Okay, and this is nothing but a combined perfect echo with G-SERV, analogous to our previous experiments. This also gives JHH for a selected proton. What is a perfect echo element? It is very well known. It is published way back, long back. It is nothing but the two echo sequences with the orthogonal 90-degree pulse at the center. Of course, you can use a hard pulse or a soft pulse, make it selective or non-selective. Okay, this is the simple perfect echo block. And using this perfect echo block, this experiment was designed called PE-SERV by this Chinese group. And quickly, I'll tell you what is going to happen here. And this block excites different spins in different slices. No more than two coupled protons are affected in the same slice here. And these two blocks refocus all the J coupling, and this will reintroduce the J coupling. Very simple, which is easily understandable. There is nothing much to explain here. But if you go to the example of a three coupled protons, what happens? Look at this one. Let us say I have two protons, I1, I2, and another proton, S. In this block, proton 1 coupled to S, proton 2 coupled to S, both are refocused. But the coupling among proton 1 and 2 is always remains unaltered. Whereas the 90-degree pulse reverses something, changes the terms. And in this case, what will happen? It will remove the unwanted J coupling between proton 1 and 2. This is the advantage of this. As a consequence, the sensitivity will be gained. That is what this sequence, which is published by Chinese group, explained in the paper. But now let us see the weakness of this paper. If you look at it, this is a spectrum taken from the original paper. And we'll look at the proton 15B. Again, it is the same strychnine molecule. Looks like that it is a favorite molecule for everybody. All the time when they do the pulse sequences, we always take this molecule to demonstrate its working. And the same proton 15B, as I have been telling all along, it is coupled to remaining three protons, let us say 15A, 16, and 14. This is what we should expect, three doublets. Of course, with the enhanced sensitivity because of the new design perfect echo sequence. But look at this, what is happening? This frequency, zero frequency peak is still present. Not only that, go to the another proton 15A. The 15A is expected to couple to proton 12, 8, and 14. And we should expect this type of doublet, three doublet. Again, we have all these artifacts. Of course, in some examples we have seen there's unwanted evolution also in addition to zero frequency evolution of the axial peaks. So now again, similar to G-SERV experiment, we wanted to see how can we clean it up. In addition, now the advantage is if we clean it up, not only we are going to get the couplings and with higher sensitivity simultaneously for all the coupled protons. At the same time, what is going to happen is we'll have a enhanced sensitivity. So we wanted to see this. Of course, this was contributed by Neeru and Sandeep recently. This now manuscript, it is already review is also over. We are expecting the editor comment today or tomorrow. So this is again the similar thing, clean perfect echo, identical. PE sequence is known, and we again started with our appended block like this. And of course, rest of the things are simple experimental details. Without going into the details of this, this is what is important. Like in the conventional previous experiment, which I explained, the delta has to be optimized by experimentally for different molecules. We did all those things. And again, apply this on a molecule like this. simple molecule, L-menthol. You can see now the difference between PE surf experiment. This is our clean PE surf experiment. In the PE surf experiment, you have this unwanted evolution. Because as I said, this proton is coupled to only remaining 3 protons. This will confuse you. Now you know you will have to start understanding what are these coupling coming from? What is this due to? Which is it coupled to? So this type of thing will not help you in identifying the true coupled proton, and also we cannot expect accurately the coupling information. So this is 1% and also presence of 0 frequency peaks. This is basic issue which is present in PE subsequence. In our clean PE subsequence, everything is cleaned up, and very precisely we are able to measure coupling directly from the separation in the indirect emission. So all the 3 coupling without an ambiguity we could measure. Okay, go back to the next bigger molecule. Again, as I said, strychnine is our favorite molecule. When we start doing that, in the PE surf, we have unwanted evolution like this. At the same time, we have 0 frequency peaks. Now, this is the clean PE surf. Fantastic. Everything is completely all unwanted artifacts are removed, and we get 3 doublets, which for this 15B coupled to 3 other protons, very precisely, accurately, we were able to measure the coupling information. Okay. Again, as I said, in the reaction alkylates and some reaction mixture, combinatorial mixtures, sometimes we come across multiple molecules present in that, in a molecular mixture. So to test that, we took the mixture of 3 amino acids, leucine, valine, and isoleucine. We concentrated on this proton of the valine, and in the PE surf, you will see lots of artifacts and evolution present. And we were unable to get especially in this region, to get the coupling, which is submerged in these huge artifact peaks. Whereas in the clean PE surf, we were able to measure everything very precisely, especially these 2 protons, 4 and 5, which are so closely spaced. This we are able to get, and without any difficulty. Again, we did the sensitivity comparison for this PE surf with our clean PE surf. Of course, PE surf, when we did the similar experiment and identical conditions, if I take this as for comparison, PE surf intensity, if I take it to be 100%, and clean PE surf is only 75%, there is again a sensitivity penalty of 25%. But remember, compared to the G surf or clean G surf experiment, because of the perfect eco-editing, there is a general enhancement in the sensitivity, like in PE surf. But compared to PE surf, it is less, but there is a general enhancement in the sensitivity. This is the thing which I say I wanted to tell you about the advantage of the clean sequences. It is free from axial peaks and unwanted evolution. It permits the determination of spectral parameters from complex spectrum in an orchestrated manner. I can get it. And sensitivity is also addressed related to PE surf, related to other clean G surf and G surf sequence type experiments. Of course, related to PE surf itself, there is a sensitivity penalty. Of course, one thing which we have not addressed here is we have addressed sensitivity, unwanted evolution, and axial peaks, and simultaneous detection of all the coupling. But what about the time? Because this is a 2D experiment. 2D experiment, as I said, because it takes time. Can we make this into a one-dimensional version of these things? I don't want to go into the details because this title of my talk is only clean sequences. But just to tell you, we also came out what is called a QG surf experiment called Quick G surf, which is nothing but the one-dimensional or the two-dimensional G surf experiment. And of course, with this, I will tell you, this is a simple example to show, and a simple molecule like propylene carbonate is a conventional spectrum, and this is a pure shift spectrum. Here, selectively, we can reintroduce coupling for a particular proton. This is called similar G surf experiment. It's a one-dimensional one. That's why time consuming is about few minutes compared to several minutes in the 2D. That's why we call it as a quick G surf experiment. And this quick G surf experiment and a bigger molecule like strychnine, again, you can see that this is the conventional spectrum, and this is pure shift spectrum where there are no coupling information present, and selectively for the same proton 15B, couplings have reintroduced, whereas all other protons are pure shift. There is no coupling. It is pure chemical shift you will measure. But only for the proton 15B, which are other 3 protons which are coupled, you are going to get the doublets where you can directly measure the coupling. So this is a one-dimensional version. This is part of my talk, but anyway, just to say, we could also address time in addition to sensitivity and other issues. So these are something which I wanted to discuss. And before I thanking the people, I wanted to tell, this is the responsibility given to me by Jeet. They asked, and Vivek, they asked me to show this in the last slide. The next edition, who are the speakers? The next edition speakers are Professor Ranbir Das from NCBS Bangalore and Professor Albert Thomas from UCLA, USA. And they are monitoring, of course, Ranbir Das will be talking on monitoring protein ubiquitination in real-time NMR. And Albert is talking about NMR spectroscopic imaging theory and selected applications. So this is, and of course, please note down the change in the timing. In this series, in first edition, because Rams had a conflict with the other webinar series which he was giving talk, and then we had to reschedule for the convenience of him. Now, in the next series onward, preferably we want to keep it to 7:30 and 8 so that it is convenient for most of the people at different parts of the globe. This is what our time which we are fixing. Anyway, we can always reiterate depending upon the need at any given time. So with this, I would like to thank all of you. Thank you very much.
Thank you, NSP. Now the talk is open for discussion. Any questions?
So there is a question from Dr. Avik Majumdar, sir.
Okay, go ahead.
He's asking, how does one take care of longitudinal diffusion during the G-surf experiments?
Yes, that's a good point, but it can happen. We do not know, but the time what we are going to delay, what we are using is very small, about, as I showed you, about 20 or 30 milliseconds. It can diffuse. That is maybe the reason why it causes also a sensitivity penalty is also there. That part we do not know.
And so there is another question from Dr. Joshi. He's,
Go ahead.
we use clean G-surf experiment in mixture of small molecules.
Pardon me, repeat the question.
Can we use clean G-surf experiment in mixture of small molecules?
Okay, we can do, that's what I showed, you know. Clean G-surf experiment in a mixture of 2-3 molecules I took, and I showed that.
Okay.
I see 3 molecules I took them, I don't know, the big names were there. In that I showed that it is possible for one of the molecules selectively, we took one proton, selectively excited and measured all the couplings. It is possible. And also in all the experiment I took, we took combinatorial mixtures. If you, I don't know, should I go back again and show you?
Not really. You can say that it's possible to have mixtures of molecules and do so.
Yeah, it is possible. I did that.
I think if Dr. Joshi is satisfied, that's fair enough. I mean, if he can follow up.
Yeah, it is clear to me. Thank you very much.
Sir, there is another question from Dr. Maya. What will be the highest molecular weight or big molecule that we can apply to these pulses? I mean,
as you see, we have seen only small molecules. The maximum size of the molecule we saw was only strychnine of that size. So we have not gone to very big molecules. Of course, there is always problem if a molecule is too big with too many protons present. Selective excitation could be a problem because of overlap. One of the requirement is the particular, at least one of the proton should be isolated for selective excitation.
Yes.
Otherwise, you will be disturbing the neighboring spins also, and then again it will complicate the system.
Yes. Any further question?
okay, I have just a query, sir.
Yeah.
So you said you showed gradient direction, right? So was this gradient paired? I mean, you're using the coupled gradients like positive and negative together, so that
IP. Now what you're telling is IP-AP sequence.
Uh-huh.
No, we have not done that.
Okay. Okay. Yeah. Any more questions? Then you can write or raise your hand.
Okay, so if there are no further questions, let us thank both the speakers of the day, Professor Ramamurthy and Professor NSP, for their, starting off the webinar series. And we are extremely excited about this webinar series. When people talked about it, we were not very sure whether it will work, but I think right from the first pair of seminars, we know that it's going to be a very working proposition. We can include later on even special topics of interest to students, maybe some learning topics. And therefore, I think this is, to me as the president personally, it is very exciting. It's a nice, every week we will have 2 lectures and people can suggest to the NMRS office whether there is any special topic in which they need a seminar, then we can find out an expert in that field and request them to give a talk. So please note that there are 2 talks coming next Tuesday, Ranbir Das from Bangalore and Albert Thomas from UCLA. And this will be starting at 7:30 and not like 6 o'clock today. So with this, once again, we actually pay our respect to Atreya and we are starting the seminar series in NMRS dedicated to Atreya. And I thank NSP, our coordinators, Vivek, Chuk, Jitendra, and everybody in the NMRS office for having made these preparations, and it went through without any hitch. So I think we are okay. There was absolutely no flaw, audio, video, everything was quite acceptable. So with this, let me thank all the audience for their presence. And once again, thanks to the NMRS office people, including Lagotama and other people. And let us hope that this is a successful venture. We just started today. Let us see how it goes. And thank you very much for all of you.
Yeah, Professor Subramanian, one thing is we have this Zoom or everything. Vivek is an expert. He did a lot of things. We should especially thank Vivek and Jeet. They are the people. These 2 are the brains. These 2 brains did all those things.
If I have missed somebody, you please bring their name up and we will thank them profusely.
No, yeah, only these 2 are.
Chog and Vivek Tiwari did a lot of work, background work.
Yes. Thank you.
No,
I'm happy.
Thank you. It was really nice, sir. Thank you.
Thank you. Thank you. All right.
If Jitendra wants to say something, go ahead.
No, I'm
Vivek
good.
also.
No.
Vivek, you have something to say for next week? Nothing.
No, I mean, I'm just I'll thank you definitely because you were guiding us, like Jiten and myself from day one, and whatever we did, actually, it was just your guidance. We were just following your suggestions. That's all.
Oh, thank you. Very kind of.
And next week we join at 7:30, and we are looking forward. We have a great list of speakers lined up till November.
Yeah.
And whosoever is interested, and they can volunteer themselves and suggest the dates that are suitable to them, and then we'll try to put them in spots. Yeah.
So, I must say that whenever we had a question or any query from NSP, like he used to answer. So the first answer from him used to come at 3 a.m. in the morning. So he was like very prompt.
That is my habit, Jeet.
Yes.
I get up at 3 every day.
Yeah. So he was surprised to see when I get up and then I see, oh, the first reply has come at 3. Yeah.
Okay.
Okay.
Thank you. Thank you, everyone.
Thank you. Okay, let us meet next week. Thank you all.
Very nice evening to all. Thank you.
Yeah.
Thank you very much.
Thank you.
Hello. Hello. Hi, Jeet. Yeah.